right. Next section, let's look at bottlenecking, which is another evolutionary implication and mechanism. So we know that over time, mutations will build up, and each new copy of a mutant lineage will probably contain more rather than fewer mutations than its predecessor. And so everything is constantly changing, and the ratchet, Muller's ratchet, is clicking one little notch every time, inevitably leading toward our demise. One thing that can help this along is called a genetic bottleneck. And so a genetic bottleneck is simply this. If you take an initially diverse population and then something bad happens, think meteorite, think plague, think economic collapse, think bleach dumped into the toilet, killing off all the little E. coli's that are there. This is a bottlenecking event because, more or less at random, you're going to select from a diverse lineage one particular variant. Maybe that's the only survivor out of this entire group of genetically diverse individuals. And now it can still replicate. And so over time, you'll get a lineage that's going to start out with these as its founder mutations. So these original mutations from the one that happened to go through, even though they may not be good mutations for the individual or for the species, these are now basically baked into the genome, and good luck trying to get them out through random mutation. There's not a good way. And now you also lost the entire pool of other individuals who you could draw on to through recombination to maybe swap regions and get rid of any sort of bad parts. Yeah. So, with a bottleneck, you will get an accumulation of mutations, new mutations plus all the old ones. And bottlenecks do lead to decreased replicative fitness. So this is what Darwin's talking about in On the Origin of Species when he's saying that no species goes extinct all at once. It's always a gradual decline. And as you get fewer and fewer individuals, it becomes more likely that eventually the whole thing is just going to collapse. They didn't have a concept of genetics that was working at the time, but we do now, and bottlenecking is definitely a reason that would explain something like this. As you get fewer and fewer members, you've basically randomly picked off a couple of genomes, and these genomes are being selected at the level of like poachers, or was there a drought in a particular, forest one year, and so this individual survived, but not necessarily through the quality of its genes. A large part of it is just random chance. And so they've actually done these experiments with viruses. So there's VSV again. It's a nice small virus, easy to work with. 5-6 bacteriophage, even smaller, even easier to work with. Foot and mouth disease, HIV, and the MS2 bacteriophage. So they subjected these things to bottleneck passages where you build up diversity and then you select it down to an individual. One of them gets to go through. And they did this multiple times. So they went from tons of virus down to just pick a little bit out and put you on, whoever you are. And over time, they then took the ending virus and the starting virus, and they compared how many copies can each of you make given the same number of cells to replicate in. And you had an average decrease, and they repeated these things several times, of like 90% fitness, 60% fitness, 20% fitness. Yeah, bottlenecks decrease fitness of the species. And there's not a lot that you can do about that. So that's also part of the concept behind quasispecies. And all of these are things that seem like they should make quasispecies not work, and yet they're going to come together at the end, and they're going to make quasispecies really work. You're going to be surprised, yeah. Here is a different one. This is Porpita, a little blue button jellyfish. You get these on the East Coast, and I think they've washed up on the beaches by Galveston occasionally, just because it's beautiful. So remember, fitness, ability to produce the largest number of given of progeny in a given environment. Fitness is going to be dependent on that environment. You will be fit for a particular situation. Nobody is fittest for everything. Usain Bolt is fantastically fit when it comes to running in a particular way on a particular track. Is he the best at calculus? I have no idea, but chances are there's somebody out there that is a little sharper at calculus or a little better at hammer throwing than Usain Bolt. Yeah. Hence, there are different Olympic sports which have different winners and different losers in them. Yeah. And so, wrapped up in this, there is this idea, and this at first just seems insane, but it actually ends up working. The idea is that we make mutations not as errors, but because we have to. As the situation changes, an individual that is best suited to one situation will not be best suited to all situations. As you go from night when it's cold to morning when it's a little warmer, some individuals are going to be better at reproducing in morning versus night versus day versus in a particular job, in a particular place, in a particular time, with other particular diseases or economic drivers around. Yeah, each of us faces a constantly shifting fitness landscape all the time. And so, if our genomes were static and they could not change, then as the environment changed, individual fitness would necessarily go down. And the fitness of the species as a whole could only go down. Because if you're best adapted to one situation, as the situation changes, you are not going to be best adapted to that new situation. Yeah, that's just how it is. Yeah. So there is, therefore, a thing called an error threshold. And there's like a desirable amount of variation within a population. There's a point where variation will increase fitness by decreasing fitness right now, but increasing potential future fitness as the situation changes. And so the optimum error rate is always going to be something higher than zero. Zero is never the optimum because nothing ever stays the same except perhaps the cold vacuum of space. But even there are different particles wandering through that space from time to time. And yeah, even if you could imagine a creature that evolved in that kind of environment, there would probably still be a non-zero optimal mutation rate. So mutations are mostly deleterious to the individual right now, but they can be necessary for the survival of the group. And individual fitness will go down with each error that you put in, like Mueller's ratchet. Click, click, click. But the greatest fitness for the entire group, the species, is going to be when mutation rates approach, but don't go over, a certain level, which we can call the error threshold. And you'll have gradually increasing fitness up to the error threshold. And then when you get too many mutations, you will get decreasing fitness down on the other side. So it's like an error optimum rather than error threshold, but that's the words that people use, error threshold. We can actually see this in play in viral genomes. So this is from some work that was done with poliovirus and done with vesicular stomatitis virus, good old VSV. So they took viruses and they compared them and saw, how many mutations they have compared to the starting genome over time. And so some had 0, but the average looked like it was around 3 mutations. And then they took these things to see whether or not they were actually fit. And all the ones that are white are viruses that could replicate to some extent. All the ones that are green are viruses that, all the ones that are over this line are viruses that were dead. And so they were able to figure out that the error optimum, the error threshold, where you go from viruses that are mostly alive to mostly dead, is going to be somewhere around 4 mutations for poliovirus. Yeah. And the error threshold for vesicular stomatitis virus, or actually, no, this is still poliovirus, but with a enhanced fidelity here. So this is there we go. And so this is the low error version of poliovirus, and it's not optimal. Yeah, it just makes fewer mutations. And so the question is, if I have a normal poliovirus, wild type, that'll make an average of, this many mutations and get me as close as possible to the 3 mutation diversity within the cloud. So your average virus is going to be about 3 mutations away from whatever the genetic center of that cloud is. Over here, you have a virus that ends up making very few mutations. It's the same virus. There's only one genetic change, glycine to serine at position 64, the polymerase. But the question is, what's the difference between these two? If you take an equivalent number of these 2 viruses, which one will be fitter, better able to survive? And the only way to answer questions like this is through experimentation. This is science. These aren't thought experiments. And the answer is going to be that the one that can change is going to be fitter than the one that can't change. Even though they start out nearly identical, mutations actually are, in the long run, beneficial. So we get this concept here, where it's not survival of the fittest, which is sort of classical Darwinist idea, but perhaps survival of the flattest. So if you are clustered at a little fitness peak like this, each individual can be nearly identical and very high fitness, but mutations will happen. And when mutations happen, they're going to push you way down the slope. So if you've got an individual on a very steep fitness slope, the idea is that it will be fairly unstable, and in the long term, it's going to have a hard time staying there. If you take something else, where it has a lower overall fitness slope, as mutations happen, it's going to move, but there's a very slight decline in fitness with the movement. And so you're going to end up with a population that, on the whole, is both diverse and just about as fit as the original. Now, you could also imagine something like a giant bubble where it has extremely high fitness and extremely high flatness. Yeah, that would be a real doozy. And so this is the concept of survival, not of the fittest, but of the flattest fitness landscape, in that it's going to be error tolerant. This is what you find in a lot of viruses because they mutate fast. They mutate faster than anything else. And if they only mutated and they could not tolerate these mutations, perhaps better than you and I could tolerate them, they would be dead every single time. So it's kind of a group evolution concept that if you have the flattest fitness landscape that your quasi-species will naturally spread out on, then you can achieve a higher average fitness for each member of the group and a higher total fitness for all the members of the group together than you can achieve if you had one very high but sharp fitness peak where everything could exist. And this has actually experimentally been demonstrated with poliovirus. They took those 2 viruses that we showed before, the ones that can make lots of changes and operate very close to their error threshold of about 4 mutations. And they took the same virus, but with a change, meaning that it operates right down near making exact copies. And they put the same amount into mice. And they found that the virus that could change could actually infect the mouse and go from the mouse's intestines into its bloodstream, from its bloodstream to its muscles, from the muscles to the nerves, from the nerves up to the spinal cord, from the spinal cord to the brain where the mouse is dead. And so it ended up being a more successful virus, was transmitted better, and was more likely to cause disease because it was able to not be diverse but create diversity. Because, of course, each of those situations is like a different Olympic event. One virus is going to be the best at getting through the stomach unscathed. A different virus is going to be better at crossing from the stomach into the intestine. A different virus may be better at going from the intestine and getting somehow into the bloodstream. Another virus, etc., may be better at the next job. And so there are actually two paths to fitness. One, either you start with a diverse population that will then help each other out along the way, or you start with one sort of medium virus that can make the entire quasispecies just through random mutation. A virus that messes up, but ends up being fitter because it messed up than if it didn't. Yeah, that's viruses. That's viral quasispecies. Here's the proof as well. So not only could the virus that could mutate, not only did that end up killing mice and being just a much more, bad virus like the regular wild type virus that made big, broad quasispecies, but they actually took viruses out of the from the wild type virus and from this G64S. That's the one that can't change. And was very close to the original. They took G64S and they artificially put in a lot of random mutations so it would be sort of broad. And they isolated each of these viruses back out of the brain. So they took the ones that were like the winners at the end of the thing, and they put them back in. And you see that all of these winners were okay at getting through the muscle, but most of them didn't make it all the way to the spinal cord or up to the brain. So the virus that was fittest at the end of the race was not fittest at the other steps that you needed to get to the end of the race, because there's no winning combination. The winning combination is that you have to make all the combinations and you have to be able to change. Yeah, a quasi-species is not about individual winners. It is about the success or failure of the group as a whole. And so we have a nice group of jellyfish in the background. I think those are maybe lion's mane jellyfish. Just to illustrate this point. Here's some of the other data from this paper, which will be posted on Canvas. So, the first step, getting through the intestinal epithelium. Here we have the wild type virus. So virus that can change are the squares. Virus that is the same in every way except that it can't change because of one mutation is in the circles. And so you see that both of these are roughly equivalent at growing in the intestine because they both start off in the same place. But can they get through into the muscle via the bloodstream to the muscle? And you see that the virus that can't change is at a disadvantage. It actually gets cleared out faster, and the virus that can change does better. Can it get to the spinal cord? The answer is pretty much no for our unchangeable virus, but yes for our changeable one. And look at that. Can it get all the way to the brain and cause death? Oh yeah, if it can change, it can do it. And if it can't change, no, can't really do it. Same virus, but narrow quasi-species in circles, broad quasi-species, or self-broadening quasi-species in squares. But do the same experiment and start out with an enhanced quasi-species where you take the one that can't individually change but manually put in lots of random changes. And so it starts out as a diverse population. The diverse population gets through the small intestine, gets through the muscle, spinal cord, brain. About the same as the virus that starts out as just a uniform virus but can make changes. So you don't have to come in diverse. You can generate diversity on site, or you can start out diverse. And either way, you can become the fittest virus in your bunch, and you can get to the brain and you can kill the mouse. Yeah, way to go. This is the heart of quasi-species theory. That groups of genetically inferior, almost certainly inferior viruses for any of the situations that they faced are together going to be more effective. And so this could be two viruses getting into the same cell, and one virus, opening the way so that the other virus can then get on to the next stage, like a relay race. It could be one virus getting in and injuring a cell in a way that makes it easier, like breaks down some sort of barrier so that other viruses can get through. There are lots of ways that this can and probably does work, and we're just starting to figure some of these out. But yeah, this is how viruses evolve. They work not as individuals, but as quasi-species. And so think of that. Next time somebody's talking about a particular virus variant, you can tell them that no, viruses may start out as a variant, but they're going to expand into a quasi-species. And it's the quasi-species that's going to get you. Each variant may have a slightly different starting point or midpoint for the cloud of angry viruses swarming around it. But yeah, really, you're never fighting one. You're always fighting all of them together at the same time. So, going back to the concept of life, was it? Yeah, this is actually my kids on the beach in England. We found a bunch of jellyfish that washed up, and they decided to make the jellyfish hospital. There's a little hole in the sand where we got the jellies and popped them in there, and they kept refilling it with water from the little buckets over here. You've got some big moon jellyfish, Aurelia aurita, and you've got some little pleurobrachia. They're called comb jellies. That's these little tiny things. They're also called sea gooseberries over there. And on this beach in the south of England, it's pretty much the same story as you get way back in time. These are from the Cambrian period, 500 and some million years ago, and these are jellyfish, much like the modern ones. They've washed up on a beach with little ripples on there, no footprints because there are no people at this time. And there they are, jellies that died on the sand and jellies that are in the jellyfish hospital. So, yeah, life. I don't know, it's not the rocks. It's not the water. It's like the action of the waterfall. It's this temporary thing, but as long as there's always water going over the waterfall, you get this constant evolution, this constant change. And so it's not the one thing, it's not the other. It's kind of what's in between. At least for me, that is the meaning of life. So there you go. This has been virology and quasi-species. And I hope you got something out of this that you can use. Because these principles apply not just to viruses. Viruses are simple and they're fast. Viruses go through all the options faster than any other kind of living thing. And so they show us what's possible. And virology is, in that sense, very much the science of the possible, the science of the quick, the science of the rapidly evolving, the science of what life could be rather than what it is, and the science of where we might be going, and who might get there and who might not get there. It's an interesting field, and I hope you enjoyed the class, and I hope that what you take from this class is something that you can use, like a little yardstick that you can compare other things to in the future. I hope you carry concepts like quasi-species, like the RNA world, like viral evolution, like cooperation, like polyprotein processing as a way to unpack things. Yeah, carry this with you, use it where you will, or just maybe be a little more interesting at the next dinner party you manage to get to. I've enjoyed having you here, and I hope you liked it. And now, it's over.